Welcome to this key area 3, looking at gene expression. There's a number of videos in this key area. This is quite a big topic, and this one is going to start off with looking at the structure and the function of RNA. So the title of this being gene expression, what do we mean by that? Well, it's the process by which genetic instructions are used to make proteins. So it's protein synthesis. When a gene is expressed, it's making a protein. These proteins can go on to perform essential functions such as enzymes and hormones, etc. Now, only a fraction of the genes in a cell are actually expressed. In other words, only a fraction of those genes actually are making the proteins. And what genes are expressed is dependent on what the cell's function is. There are genes that are switched on in order for them to be expressed, in other words, for them to produce proteins. And there are other genes that are not used, that are switched off because they're not needed for that cell's function. Now, this gene expression involves processes called transcription and translation of DNA sequences, and we'll go on to have a look at what we mean by that in another couple of videos. But RNA plays a vital role in the production of proteins, and there are three types of RNA. There's messenger RNA, and you may remember that from your National 5. There's transcribed RNA, and you should remember that as well, TRNA. And there's ribosomal RNA. So let's have a look at the structure of RNA. Now, RNA nucleotides have got a similar structure to the DNA nucleotides. But there are a couple of differences. The deoxyribose sugar in DNA is replaced in RNA with a ribose sugar, and the bases are the same: adenine, cytosine and guanine, except thymine is replaced by a base called uracil. You don't need to know the structure of ribose, but this helps the ribose sugar. This just helps you to see the difference. You'll see the ribose sugar has got an OH group here on the ring, and the deoxyribose has got a hydrogen group here. So, a summary of the differences: DNA is double-stranded, RNA is single-stranded. DNA has a deoxyribose sugar, RNA has got a ribose, and the bases in DNA are A, T, C and G, whereas in RNA there are A, U, C and G, and the uracil there is replacing the thymine. Messenger RNA is formed in a process which is called transcribing in the nucleus from free nucleotides, and you'll remember, or you should remember from your National 5, it carries a copy of the DNA code from the nucleus to the ribosomes where protein synthesis occurs. That's a process called translation. Each triplet base of the mRNA molecule is called a codon, and it codes for a specific amino acid when that mRNA molecule gets to the ribosome. So that's just a brief reminder of what messenger RNA does. So your tRNA molecule looks something like this diagram that is over here. Instead of just being a straight strand, what happens is that the strand folds up because of complementary base pairing. In other words, you've got your long strand, and what happens is some of that long strand is attracted to, because of base pairing, to another part of the strand, and you end up having this folding situation happening, which gives the tRNA molecule this specific shape. Now, each tRNA molecule has a specific amino acid attached to it, because if you remember, the tRNA transfers the amino acids to the ribosome depending on what the order is of the codons on the mRNA molecule. And at the other end of the tRNA molecule, you'll see this anticodon, and that is a triplet of bases at the end of the tRNA molecule, which are complementary to the codon triplet of bases that is on the messenger RNA molecule. And this is your third RNA molecule, which combines with proteins to create the ribosome. That is the organelle responsible in the cell for assembling proteins. Now in the next couple of videos we're going to go on and have a look at gene expression. So a gene is a section of DNA which carries the code for the production or the expression of a particular protein. And this process has got two stages, transcription and translation. So in the next video we'll have a look at the stage of transcription.